our seminar speaker. He is the Terry Presidential Professor at the University of North Florida in Jacksonville, where he also serves as the chair of the Department of Biology. He's been a faculty member at UNF since 2007 and has advanced through the ranks there while receiving lots of awards and recognition, including an Outstanding Faculty Scholarship Award, an Outstanding Undergrad Teaching Award. Love to see those in seminar speakers. It means we're in for a good seminar. A Disability Resource Center's Empowerment Award and the Dean's Council Faculty Fellowship. He, in addition to his UNF position, is also a research associate at the Smithsonian Marine Station, where he had previously done his postdoc. And prior to that, he earned his PhD at University of California, Santa Barbara, and his bachelor's and master's degrees at Florida Atlantic University. Cliff's research broadly focuses on ecological physiology and understanding stress responses in a variety of species, including seagrasses, corals, and algae. And I'm particularly excited to have him here with us today because I recently reached out to start a collaboration with Cliff on looking at disease in seagrasses, and he's going to tell you about some of that work today, specifically with seagrass wasting disease in Florida, current insights into a Labyrinthula turtlegrass pathosystem. Thanks so much.
Okay.
Take it away, Cliff.
Yeah, thank you very much, Maya. Let's see if I can do this successfully, and please let me know if you can't hear me or if it's not working out. And let me move everyone else around. Okay. And I just learned how to use the laser pointer option. Look at that. Okay, can you guys see my slide and my laser pointer?
Yep, we're good. Slides, laser pointer, and volume. So I'm gonna mute myself now. You're good to go.
Okay, thank you so much. Great, super. So let's kind of advance on there. I kind of wanted to reiterate my academic trajectory and my travels. I just went through it all, but I just kind of wanted to use Google Earth. I never really use it, so let me use it for this. But as Maya pointed out, I'm from South Florida originally. I grew up in Boca Raton, Delray Beach area. For my undergraduate studies, I attended Florida Atlantic University, got a degree in marine biology, and then I stayed on and did a master's degree in marine biology as well. And then I traveled across the country to the University of California, Santa Barbara, and actually overlapped with Amelia there. And I still look back at that. That was definitely one of the best experiences of my life. I loved it out there in the interdepartmental program in marine science. Really phenomenal. I wanted to get back to Florida, though, just because a lot of the research I do deals with tropical or subtropical entities dealing with corals and seagrasses. So I was able to get a postdoc after my PhD under the guidance of Valerie Paul, the director of the Smithsonian Marine Station at Fort Pierce. Again, great facility as well. I loved my time there. And that was from 2005, 2007. And then at 2007, I was able to land a position at University of North Florida, as Maya pointed out. And that's been great. UNF is a relatively young university, and it's been really fun and exciting to help kind of shape the trajectory of the department. And here we are today, just kind of chugging along. If you ask, what do we do or what do I do in my lab? I'd say we dabble. We do a little bit of everything. Probably the best description I can offer is I would classify it as integrative ecological physiology, where we take physiological approaches, cell and molecular approaches, and of course we couple that with field work, the fun part, to address questions pertaining to stress responses in corals, seagrasses, and also we do work with microalgae and macroalgae as well. In the most general sense, we ask questions like, how do marine organisms adapt to a rapidly changing environment? Or how do marine organisms respond to each other in the context of this rapidly changing environment? Okay. But today we'll be focusing on diseases, specifically seagrass diseases. And, if you look in the literature, I think you'll find quite a bit out there demonstrating that emerging diseases and sometimes even re-emerging diseases that are coming back at us to a certain extent are correlated or are associated with climate change and anthropogenic activity. And while the importance of pathogens in terrestrial ecosystems has certainly long been documented, the role of diseases in marine habitats has simply just not been as well as studied, as demonstrated by the paucity of information that's out there. And so where I'm going with this is that increasing our knowledge of host pathogen interactions in the context of this changing environment is quite important to understanding the future trajectory of coastal ecosystem health. So when we talk about diseases, and let's use the example of seagrass diseases, we have this disease triangle. I think a number of you have seen this before, where you have this equilateral triangle and you're dealing with three major components: the health of the host, or how the host is doing, the pathogen, and of course, right, the ever-present environment, which is kind of serving as the mediator, which will kind of dictate the balance and who's going to be the winner or loser in a certain situation. And even though we have these three macro components, things can certainly get complicated pretty quickly once you want to consider all the subcomponents that build these three macro components. For example, when we talk about the environment, right, you're concerned about biological factors, who's eating who, or in the case of seagrasses, who is grazing upon who. We certainly care about the physical factors, right, temperature, salinity, pH, chemical factors, pollutants, or in the case we'll be talking about today, nutrients, nutrient loading in a system, right? When you talk about the pathogen, we care about the pathogenicity of the organism. Some organisms are opportunistically pathogenic, whereas sometimes they're pathogenic, sometimes they're not. And when they are, we care about how evil you can be, or kind of their level of virulence. Are you very virulent or not? And we care about the transmission as well. When we talk about the host, and there's many other, I'm just giving a few examples here. When we talk about the host, we care about demographics. What kind of populations are we dealing with? Old individuals, young individuals, big individuals, small individuals. The genotype of an individual most certainly matters. How's the immune system functioning of these individuals? And can they turn it on? Can they turn it off? Is it inducible? And so just by having a change in one factor, if you look down below here, let's say there's a change in a chemical factor, perhaps the introduction of a pollutant into the environment, this can trigger a lot of different things, right? Maybe that'll cause the immunity of the host to drop, but at the same time, it might cause the virulence of a pathogen to lower. And so you got a question there, what's happening? And this can certainly serve as the basis of many hypotheses. So things get pretty complex, and you can ask a good number of questions when you talk about host pathogens interactions. That's for sure. You guys get the message of this slide, I think, right? Every time, at least for me, if I talk about seagrasses and I'm teaching a class or I give a public lecture, I have to show this because, frankly, nobody cares about plants, right? But you get the idea, right? All your favorite entrees and appetizers, or at least a good number of them, need seagrass beds as they provide that structural habitat. They're vital nursery grounds. They stabilize sediment. And as a function of time, more and more assignments or functional roles are being assigned to seagrasses. This was an interesting paper that just came out a few years ago by Lamb et al., where they demonstrated that seagrasses can serve as biological filters, specifically absorbing up pathogenic bacteria in the environment. Briefly, they were looking at islands, remote islands in somewhere off Indonesia, and they found that islands that had a seagrass bed around them really had a significant reduction in pathogenic bacteria, disease-causing bacteria like Enterococcus, right? Whereas other islands that had no seagrass beds, they saw an increase or a prevalence in these damaging bacteria. So very important, that's for sure. I'm almost embarrassed to talk about Florida Bay when I saw some of the people coming into the chat room here, or the meeting, when I see people at FWC. So if you want to ask details about Florida Bay, you can ask the FWC folks. But for the context of what I'm talking about, I can give you some of the highlights, that's for sure. Some of the work we've been doing occurs in Florida Bay. If you don't know where that is, it's on the bottom of Florida, and it's a relatively shallow water estuary. I believe in size, it's about 2,200 square kilometers. And the important point, which I was kind of naive to, but I just really learned about this in the past several years, is the fact that you have these interesting mud banks throughout the place that cause this basin, if you will, to be subdivided into miniature basins. And some of these have their kind of own ecophysiographical properties. So for example, salinities can differ here versus here. Turbidity can vary in different places. The constituents of the sediment can differ. Subcomponents of the nutrient pool, like nitrite versus phosphate, can differ in varying areas. So it really sets the stage. It makes this nice comparative system if you want to look at the effects of, say, different environmental properties or different environmental stressors on an ubiquitous organism like turtlegrass that you would find all over the place. Okay. And as the FWC folks can tell you, there's been some problems with the seagrass beds in these areas. From my understanding, in the past 50 years, there's been at least two significant die-offs. I believe one big die-off of seagrass occurred. It was 1987, excuse me, lasting through 1991, and then more recently in 2015. I think in the one back in 87, about 23,000 hectares died off, causing downstream effects. Local shrimp populations kind of plummeted, with also causing further impacts on the fisheries. What caused it? From my understanding, most of it is due to abiotic factors. Thermal stress, warm temperatures, coupled with a lot of evaporation, so high salinity, you're getting stratification, right? All of a sudden you start to have sulfide buildup coupled with hypoxia, in some cases anoxia. You have these big die-offs in the area. And there's been a few papers talking about this, going back to this one, right? So this was a report of the mass mortality going back in 87 through 91. Something that caught my eye was this one statement. It states, a pathogenic protist related to the causal agent of the eelgrass wasting disease may be involved in the mortality. Backing up just a second, eelgrass wasting disease. In the 1930s, there was a huge die-off of temperate seagrass, not in turtlegrass, but in Zostera marina on both sides of the Atlantic. So if you think of North Carolina pushing further north up through Maine and Canada, and then on the other side of the Atlantic, UK, Spain, Portugal, France, about 90% of the Zostera died off due to this disease, this wasting disease. Okay. And so there was a suspicion that perhaps this could have something to do with the trouble we see in Florida Bay to a certain extent. All right. This is the culprit. This is Labyrinthula spa. So there's a number of different types of labyrinthulids that are out there. They are eukaryotic microbes, about 10 microns in length. They have this spindle-shaped appearance. They look like flattened, deflated footballs. And they're mobile. They move around from point A to point B through this ectoplasmic network, and they move by this actin-myosin-based process. Okay, most of them are perfectly fine. I mean, ecologically speaking, they're like fungus. They're like decomposers. But some of them are pathogenic, and that's the big question, trying to discern who's pathogenic, who isn't, who might be opportunistically pathogenic. For those of you that like, phylogenetics and taxonomy, where do these guys go? That's a great question. For a number of years, they were lumped together with fungus, but they're not fungi. They don't really have chitin. Then they were grouped with slime molds, but if you know what slime molds are, they're not slime molds. So now we put them in this filing cabinet we call stramenopiles. It's also known as heteroconts. If you're not familiar with that, it's kind of like a miscellaneous filing cabinet. The common feature of stramenopiles is that at some point in your life, you've got this biflagellated zoospore phase. It's widespread. I mean, diatoms fall in this group. Brown macroalgae like kelp fall in this group. So we can put Labyrinthula in there as well. It's kind of like if you wanted to organize your garage, you can put your screwdrivers over there, your hammers over there. And at some point, you're left with a bunch of nuts and bolts, and they're all kind of look like they're chrome. That's good enough. Let's just put them in this one drawer. We'll call that drawer stramenopiles, really. All right. So symptoms of wasting disease or signs of wasting disease. Oftentimes you'll see these brown-black necrotic lesions. And one point I want to kind of plant the seed now, and we'll keep coming back to, is that just because you see lesions doesn't mean it's disease. Many things can cause lesions. Desiccation, natural senescence, etc. But this picture you're looking at on the left is particularly suspicious just because it looks like you have such vibrant green, healthy tissue on top, a lesion, more vibrant green tissue, another lesion. So clearly something's going on right here. Sometimes the situation presents itself with these streaks you can see, right? I've looked at hundreds and hundreds of samples. Probably one time, I don't have to say probably, one time we saw Labyrinthula with a microscope. This is that photograph, but most of the time you just can't even see it with the eye. This is taking kind of a longitudinal cross section, looking at some of the epidermal seagrass cells. So we're looking at 3 seagrass cells. Inside, this green stuff is just degraded organelles and chloroplasts. But you can see these kind of translucent guys. Here's Labyrinthula in there, but very difficult to find, at least in my opinion. I know years ago, there's been studies in Tampa Bay and elsewhere where some people were able to see things more effectively and efficiently. Okay, so what we do is we travel around to different places, and we'll get to a global perspective in just a little bit, and we can actually take seagrasses and other hosts, and you just plate them out on this nutrient-rich agar, and Labyrinthula will start to grow out. That's this flocculent material. You're going to get a lot of other stuff as well, fungus and other things, but using serial dilutions and isolations, you can start to get this nice monoculture. We've got a number of cultures operating right now. Now, as I mentioned, one of the biggest die-offs occurred in the 1930s. That was like the big report of wasting disease, where about 90% of the zostera was lost in the Atlantic basin. And even though it's not very famous, if you look in the literature, there's been other reports of die-offs that have been going on for quite some time. But my point of showing you this map is that we really don't know a lot in terms of host-pathogen interactions with respect to Labyrinthula and seagrass in turtlegrass populations in Florida, the Gulf of Mexico, and even the wider Caribbean for that matter. All right. So even though Labyrinthula can be considered the most well-known seagrass pathogen, and that's, to all 9 of us who study it, we really don't know that much about it. We really don't have a good grasp on the number of species that are out there. Again, it's not only on seagrass. You can find this stuff on detritus. You can find this on mangrove leaves, spartina, different types of algae. It's all over the place. We don't have a good handle on the mechanisms of dispersal and infection. We really don't have a good grasp of the species distributions and the host specificity. Hopefully, in just a little bit, I'll be able to shed some more light on this, but nonetheless, we really don't have a good handle on the factors contributing to host susceptibility or the factors contributing to pathogen viability and virulence. Let's talk about that. So let me kind of give you a run through of what we've learned over the past few years and show you where we're headed. And let's start on the plant defense side. I had a student some years ago that was interested in looking at plant defense properties. And one thing we were interested in is looking at the roles of reactive oxygen species, which is found all over the place. They're ubiquitous. If you're not familiar with reactive oxygen species, you can kind of think of them as the unwelcome companions of aerobic life. Ever since oxygen started to form in the early Earth's environment, we had the production of these reactive oxygen species, where if you take molecular diatomic oxygen and sequentially reduce it, you get a variety of reactive oxygen species. Some of these you've heard of before, like hydrogen peroxide. That's the most stable form that you can find. Now, briefly, these ROSs, reactive oxygen species, they can be good and bad. Bad in the sense that when a cell or an organism starts to undergo some type of dysfunction, reactive oxygen species start to build up. And it usually happens when electron transport systems fail. So if you think of a mitochondria or a chloroplast, electrons are flowing from point A to point B all over the place. But if something is stressed out, let's say there's thermal stress, proteins start to denature, the system falls apart a little bit, and electrons are not going to where they have to. They float off into an aerobic environment, and now you start to reduce oxygen into reactive oxygen species. And the problem with that is they're very reactive, hence the name, and they can oxidize proteins and enzymes. They can oxidize lipids, causing membrane instability. They can oxidize nucleic acids, and really they can just wreak havoc. And a lot of times they're used as biomarkers of stress, even in human diseases. Okay, that's the bad side. The good side is they can actually be produced in a regulated manner, and they are the first line of defense when we encounter pathogens, we use reactive oxygen species as well. I mean, when you hurt yourself, right, you can clean out the area with hydrogen peroxide, same concept. But when you're exposed to a pathogen, some of your cells, macrophages and neutrophils, will have a burst of reactive oxygen species, which can try and help, kill off the pathogen. But ROSs also serve as cell signaling agents, which we'll talk about in just a little bit, that can turn on certain defense genes and otherwise. So in this one study, and I'm just going to highlight some of the important points so we're not here all day. We wanted to assess whether Thalassia, seagrasses collected in Florida, can undergo this oxidative burst. When they are challenged with a pathogen, can they have this first line of defense? And so in this system, we've used Labyrinthula, but we've also used lipopolysaccharide, LPS. That's a commonly used compound. You find that in gram-negative bacteria. It's a constituent of gram-negative bacteria. It is widespread in the literature. It's the go-to compound to induce an inflammatory response, okay, or an immune response. It's used in animal models, other plants, higher plants, etc. So what we're going to do is expose Thalassia to LPS, and we want to assess its innate immunity. We're more advanced humans. We have adaptive immunity where we make specific antibodies and we have a memory system. We also have an innate immunity, but lower organisms, plants and metazoans like corals, they have an innate system, which is more like a general response. They respond to the infection the same way, maybe with certain cells migrating to the area, producing reactive oxygen species. At any rate, this is what we see in turtlegrass. If you expose it to lipopolysaccharide, after about 20 minutes, you get this little burst of hydrogen peroxide, and we're measuring that by using this hydrogen peroxide chemiluminescent luminol assay. Okay, get a little burst. If you take all the data points we see here at 20 minutes, we're just putting them on this graph down here. I'm just showing you the fact that the threshold, it requires about 50 micrograms per mil of LPS to have a significantly inducible response. Okay. We can also visualize that, the production of reactive oxygen species, by the presence of this fluorogenic probe. This probe turns green. It's called DCFHDA. The name, I think it's dichlorofluorohydrazine diacetate. If you take a cross section of Thalassia, and if you focus specifically on these epidermal cells, we switch over to fluorescence. All the red is just chlorophyll. The green starts to appear when reactive oxygen species are being produced. So it takes about 10 minutes to start to come on the scene. And by 30 minutes, you've got this strong response. ROSs are being produced, certainly in response to LPS. Okay. When you have an oxidative burst, there's usually a couple of other criteria that go along with that. So this is just some additional evidence. Oxygen is sucked into the system and it's converted into reactive oxygen species or superoxide anion. We see that upon LPS exposure, you produce more ROSs. As it happens, the pH of your media or the intracellular pH starts to go up and we see alkalization as well. So we're definitely dealing with an oxidative burst. Now this is where it gets fun. So you can expose seagrass to a pathogen or LPS, and it's going to produce hydrogen peroxide, which, as I'll show you, can start to fight back and kill off the pathogen, or it can serve as a cell signaling agent and start to turn on selected genes. But we were quite interested in what's going on right here. There's a black box. How does a presence of a pathogen turn on hydrogen peroxide? And so what we can do, I know this is a laundry list of terms and numbers, but all I want to tell you about what you're looking at here is we can take a series of pharmacological agents. Some of them are specific pharmacological activators. Some of them are specific inhibitors of specific processes. So for example, we can take a piece of seagrass and we can provide it with a calcium channel blocker, and then we can provide LPS, and we'll see if we get hydrogen peroxide. If we do not, we can infer that calcium channels are involved. And so what we can do is kind of run through this, and we can use all kinds of compounds to kind of dissect and tease apart what's involved and who goes first and who goes second. We're not going to go through all that, but it was really kind of fun figuring this out. And there's a lot involved in signaling an oxidative burst in seagrass, that's for sure. Okay. Moving back up, let me just point one other thing out. Oftentimes, the critical enzyme involved are these NADPH oxidases. That's what's going to take in oxygen and convert it to a certain type of reactive oxygen species. We were able to clone and sequence one of these coming from seagrass, and there was quite a bit of homology coming in alignment with Arabidopsis, this model plant organism. Okay, so we've demonstrated that, yeah, seagrass can produce reactive oxygen species in response to LPS. What I didn't show you is that sometimes it will produce reactive oxygen species in response to Labyrinthula. Some labyrinthulids don't trigger a response, which is good for the pathogen. Okay. Seagrasses are also loaded with secondary metabolites. And we kind of went through this. Most seagrasses have a lot of simple phenolics, like you see right here. And all the phenolics that we isolated and tested do show a certain degree of bioactivity against Labyrinthula. We've also gone on to purify and isolate more complex secondary metabolites, like this flavone glycoside. Point being, though, it also is bioactive against Labyrinthula. So where we're at is seagrasses can produce reactive oxygen species in response to infection. They've got some metabolites that can help out and defend themselves against Labyrinthula. But the point is, this stuff doesn't always work, right? It depends upon the environment and the stress. Sometimes they just can't mount a response. Big question is why? Okay, this is just another little picture here, a little blurry, sorry, but what it shows is here's a lesion induced by Labyrinthula. And if we use some fluorogenic probes, we're localizing phenolics, we can see that phenolic compounds are kind of shunted to this lesion area. It's like the plant wants to defend itself by getting phenolics to the area. And here's back to that probe for the ROS probe. We can see that there's reactive oxygen species being produced there as well. I had another student, Stacy Trevathan, and she was very interested in the effects of hypersalinity on these host pathogen interactions. And we would expect going into this that anytime you expose a plant to a stress, you figure the immunity is going to drop and then it's more prone to infection. But remember, we talked about that triangle, how the environment, kind of dictates what's going on. So this is a good story in watch what's happening here, because it's kind of hard to tell who's going to be the winner. When you take seagrass, these populations were actually collected from the Indian River Lagoon, so back when there was turtlegrass, and they were from an environment that was at a salinity of 30. So our ambient or our control specimens were maintained at a salinity of 30. And then we exposed some seagrass, these guys, to a salinity of 45, really bringing them up there. And what we were able to show is, yeah, the grasses that were placed under these hypersaline conditions were certainly stressed, whether they were infected or not. We show that chlorophyll, total chlorophyll content starts to drop when you're exposed to hypersaline conditions. And then we can look for reactive oxygen species. If you look over here, in panel A right here is a seagrass, just using bright field microscopy, looking at it, showing a sample that was exposed to ambient conditions, 30 parts per thousand. Move over to fluorescence. The red is chlorophyll. You can see a little bit of yellow-green indicating the presence of reactive oxygen species, which are just ubiquitous. They're always going to be there. Looking at picture C, that's just bright field, looking at the sample that was exposed to 45 parts per thousand. However, when we go to fluorescence, you can see there's a buildup of reactive oxygen species there. And we can quantify that. Looking over on the bottom left, the plants that were exposed to 45 parts per thousand, or PSU, I know it's unitless technically, but the point is you see a lot more hydrogen peroxide being produced. So the plant is stressed, and again, this is the bad sign. It is producing reactive oxygen species, probably because it's undergoing dysfunction. It's not happy. But at the same time, elevated levels of hydrogen peroxide, if we look over here, are capable of killing off Labyrinthula. So even though hypersalinity might be bad for the host, in this case, it may be even worse for the parasite. Okay. The boat on the top right certainly might look quite familiar to a number of you. I know when I show people in Jacksonville what it is, they don't know what it is, but we've certainly had some good times on this boat throughout the years. And so we went down, thanks to FIO funding, for a number of years, going to the Marquesas and the dry tortugas to try to make some assessments looking at the relationship between seagrass health and Labyrinthula wasting disease. And let me just give you a couple of highlights about that. In this one study, going back in time, pretty basic study, we were surveying areas, looking at plant abundance, looking at wasting disease prevalence, and I'll kind of, give myself a caveat. We're looking at lesions, which is not the be-all end-all criteria. We were assessing plant health through photochemical efficiency and looking at water quality and a number of other laboratory analyses. And the take-home point of this story is that when you look at leaf and total biomass, okay, and also look at carbon content in the root and rhizome, as well as light levels, all of these three criteria have a strong negative correlation with wasting disease. And this is not earth-shattering news, but what we're showing is that low light makes a weaker plant. Lower light levels probably, lower the levels of photosynthetic capability. Carbon content goes down. The plants are punier. Appears like they're more likely to be infected. But the point is, this is the first time that this really kind of factually demonstrated that, yeah, light availability is something that needs to be reckoned with. That's for sure. Okay. Now in Florida Bay, again, you're not just dealing with variable light levels. We have a multi-stress system, and the big stressors once again are temperature, salinity, sulfide, and oxygen levels. And so I had another graduate student, Nicole Bishop, who wanted to further study this in a laboratory setting. And so what she did was she set up this multifactorial experiment where Thalassia was exposed to different treatments, different salinities, different temperatures, presence, absence of sulfide, and hypoxia was introduced. And this study really had two facets. Part one, we're going to take the plants and we're going to expose them to any combination of stressors for 1 week, 7 days. After the 7 days, keep the stressors applied, but we're going to infect them, which is very important. And the pathogen has to infect under those conditions, so the pathogen is exposed to that stress as well. On the other side, the other part of the study was taking the plants, stressing them out for 7 days, just like we did in part one. But now the difference is, after 7 days, let's remove the plants and place them in clean seawater, ambient, okay, where the plants can be happy, and then we're going to infect them. So the pathogen does not have to deal with the stress. And this offered some interesting results. You may ask, how do you infect these things? So we have Labyrinthula cultures. What you're looking at here is a plate of Labyrinthula. All this flocculent stuff is Labyrinthula. Up here is just a chunk of agar. So every time you want to keep your culture alive, you cut out a piece of agar and you put it on the fresh plate. And what we'll do is we'll take a little piece of seagrass. If you see this little clipping here, take a clipping of a piece of seagrass, autoclave it, then place it on the agar plate for about 5 days, maybe a week. And the Labyrinthula are going to grow all over it. And now you just made a vector. And at that point, we can take the vector, we can clamp it on to a healthy piece of seagrass. We use Tygon tubing. You slit a little notch in it and it makes a nice clamp. And basically, you can kind of infect it under whatever treatment you want to. And in this case, after several days, this lesion started to appear. So it was definitely infectious. Okay. And so let me just kind of give you the highlight of what we saw. I'll just show you the results for plant health. And that's being monitored by photochemical efficiency. How well can you undergo photochemistry? Okay. With these data, essentially, you don't want to go below the red line. That's kind of the threshold in this case for your photochemical yield in seagrass. And the take-home point is this. If you stress the plant for 7 days, they're actually pretty tough. They can take a lot. And then if you infect them with sustained stress, so keep the stress going and put the pathogen in there, turns out the pathogen doesn't do very well. And so we can see that the plant is actually doing okay. But look what happens in this situation. If you stress the plant out for a week, and then again, put it under ambient conditions, so it's in this recovery condition, then you infect it, Then the pathogen can go to town. Pathogen is fine. The plant is still dealing with its past stress a little bit, it seems. And then we start to see kind of a repression or a depression in the photochemical yield. Okay? And this is kind of a good little summary here. If you take all the plants and expose them to a stress, and then you infect them with the stress, and it doesn't matter what the stress is, sulfide, hypoxia, salinity, you just simply don't get a lot of lesions because the Labyrinthula is just having a hard time with that. But if you have the plants and stress them and then place them in ambient conditions, then you infect them, okay? Doesn't matter what the stress was, just the Labyrinthula does very well and you get bigger lesions. So in summary, it seems that relatively short-term exposure to common environmental stressors, generally speaking, has a negative influence on Labyrinthula activity or viability and virulence that outweighs the effects of any reduced Thalassia testudinum photosynthetic health. So we kind of have this asymmetry that favors the host in this case. However, that asymmetry is kind of flipped or reversed, so the pathogen wins when the stress is removed or alleviated. Okay. And I'm coming back to the point again that when we look for disease, you don't really always want to focus on the lesions. You know, even sometimes if you infect seagrass with Labyrinthula, some of these plants might get pretty big lesions even though it's not really infected that much. The levels of infection are low because the plant has a defense response. We can find that the plants will undergo programmed cell death. It is actively killing itself off to make these dead barriers so there's no viable tissue for the pathogen to move into. So the size of a lesion can be a little bit deceiving. It may not accurately represent the pathogen load. And so to address that, we wanted to develop a molecular tool, a toolkit that could quantify the loading potential, how much pathogen we can find in seagrass. And so a colleague of mine, Dan Martin, who is kind of this Labyrinthula expert, he was collecting Labyrinthulid samples from around the globe and collecting samples all over North America, Europe, Australia. I was able to go to, when I was doing some work in Belize, send him some samples from Central America. And he was looking at their phylogenetic relationship based upon this ITS1 sequence. Okay, and so let me kind of give you the main point of what he found. It's really interesting. So you can collect Labyrinthula from all over the place, from different environments and different hosts. Remember, you can find this on detritus and even mangroves. And it turns out that if you're non-pathogenic, you can kind of make these clades that we see in white over here. Interestingly, if you are pathogenic, and it does not matter what biogeographical area you were collected from, you could have been collected from seagrass in Washington State, you could have been collected from a mangrove in Northern Australia, Florida, Belize. If you are pathogenic, you group out up here. And this is very exciting. So what that demonstrates is that a component, right, of the ITS1 sequence seems to be quite homologous. There's a lot of homology there, which enabled us to develop primers to target the pathogenic Labyrinthula. I mean, ultimately, that's who we're interested in. And so I had a great graduate student, Paige Duffin, and she worked with Dan Martin in developing this primer set where we can kind of test that. And so what they did was they took a number of isolates collected around the globe. 31% of them, or 12 of them, were pathogenic. A number of them were non-pathogenic. And as it turned out, everything pathogenic would always get a hit, which is great. Now, the non-pathogenic ones, I'll be transparent with you. The vast majority of the time, the non-pathogenic ones do not amplify. Okay? A couple of them do, right? But they only do, you know, with the following issues. Or I shouldn't say issues. We just, with the following observations. The non-pathogenic ones, a couple of them will amplify, but if you do qPCR or PCR, you will always find that the milk peak is different than the pathogenic ones. Furthermore, you can only amplify the non-pathogenic ones if you have a purified culture of non-pathogenic Labyrinthula and a high yield of that DNA. if you collect a sample from the environment, right? So take a clipping of seagrass, grind it up, which is what you're going to do, and you're going to look for Labyrinthula content. You're going to have the Labyrinthula DNA, you're going to have the seagrass DNA, you're going to have other organisms' DNA. Once there's a mixture of DNA present, the non-pathogenic ones don't amplify. So in other words, under environmentally realistic scenarios, the non-pathogenic ones don't amplify. But if one asks me, do the non-pathogenic ones ever amplify? Sure, only if they're absolutely purified in culture and you have a lot of DNA. Okay. Now, what can we do with this? Let's take this toolkit and move it back to Florida Bay. And this is where we started to team up with Brad Furman. I saw his name there, where we wanted to get some expertise of somebody that's kind of a real seagrass ecologist and that deals with Florida Bay. So we wanted to take this molecular tool and start to ask, are there site-specific factors that regulate Labyrinthula loading, right? Also, are there site-specific factors that regulate host immunity? Now, I didn't talk about this yet. I just talked about the fact that plants produce reactive oxygen species and they have secondary metabolites. There's also a number of other biomarkers that can be used where one can assess the immunity, per se, of a plant. We didn't have to reinvent the wheel, really. I mean, a lot of these enzymes and this immune panel you see here, people have established with other plants, more so agriculturally relevant plants, where there's been a lot of funding and a lot of studies looking at, say, like citrus disease or broccoli disease, things like that. But what I'm going to show you is, and if you want any of this info after, I'll be happy to email it to you. I'm just going to show you some data pertaining to these 4 enzymes shown in white: exochitinase, peroxidase, polyphenol oxidase, and lysozyme. Okay, and so we're going to ask a few questions here. Can we correlate environmental factors with Labyrinthula prevalence? Can we correlate immune responses with Labyrinthula prevalence? Right? Can we correlate these same immune responses with site-specific environmental factors? Okay. And so to do this, we're going to use these immune assays. These are all functional enzymes where we can assess how their activity. We're going to utilize water quality data that Brad was able to obtain. There's historical data by the South Florida Water Management District and the Everglades National Park. And we'll assess pathogen loading by using this newly established qPCR technique. Okay. And so here's what we see. And incidentally, this should be published any second now. It was accepted in Frontiers in Plant Science a week ago, so we just submitted the proof, so it should be out. But here's the basis of this study. This is a snapshot, mind you, so it's really kind of a one-time snapshot of what transpired. So this is going back to May 2015, where Paige was able to go out with folks running the Florida Bay Fisheries Habitat Assessment Program run out of FWC and UNC Wilmington. And so we greatly appreciate the fact that she was allowed to go with them to collect samples. And again, we went back to all these little miniature basins that have their own properties. Okay. And so to keep things normalized, Paige would go out and collect 10 samples from each site. For consistency, she would collect the 3rd rank leaf. That just really means the 3rd youngest. So not the little one, not the next one, but this one. Just because with a little bit of age, we find oftentimes you tend to be more susceptible to infection. If you're too young, you haven't had a chance to get infected yet.
How are you doing?
Okay. And so what will happen is she'll collect the samples and she would collect the leaf, cut it longitudinally. Half of it would be used for immunoassays and the other half would be used for qPCR pathogen quantification. And here's what we see. So we're looking at all these sites and we're looking at prevalence. Were you infected or not? And then severity. If you were infected, how much? So every pie chart represents a site. And every little piece of pie represents a sample. So again, n equals 10 within a site. So if you go over here to Duck Key, for example, not much activity, nothing, 0 prevalence, nothing was infected. But if you go over to Joe right here, interestingly, 10 out of 10 were infected, and the severity was rather high on our scale here. They're all turning blue. So something's kind of interesting going on up here. And so let's just put a couple of clues together. So this is clue number 1. Some sites get infected, some do not. And when they do, it gets kind of interesting, like the situation with Joe. Immune responses. We're looking at those 4 enzymes, peroxidase, exochitinase, polyphenol oxidase, and lysozyme. The larger the circle, right, the higher the activity. Exochitinase is a little weird, but if you look at the others, I think you'll see a trend, right? Larger circles are found in the northeast. So that's another clue. What's going on with that? Why do we see higher immune responses in the northeast region of the bay? Here's another way to look at the exact same data. If you want to look at all these immune biomarkers, all 4 of them, let's check them out as a function of their geography. The west side, the central side, the east side. Look how the activity is higher in all these guys in the east. Here's another observation. Looking at plants, the size. Interestingly, the size of the plants decreases in the east. There's smaller plants in the east. So let's kind of step back, regroup, and kind of synthesize this here. So what we're looking at is, if you start to look at these regions, really, we see higher immunity in the beige up here in the northeast. Okay. When you start to look at leaf area, it's kind of the opposite trend. Smaller leaves are in the northeast. And then when we look at loading, we can put that on a scale of 1, 2, 3 for our purposes right now. Seems to be most of the time the highest loading is in region 1, but you kind of have this weird dichotomy where the highest loading is right here in this coastal zone. But once you move off the coast a little bit, the pathogen levels or the severity seems to plummet. So what's going on there? And I know Paige and Brad looked at a number of different types of parameters and data, and there were some interesting findings with respect to salinity. If you look down below, we're showing you here kind of salinity trends over the course of 5 years leading up to the collection time point, which was May 2015. And what's interesting here is if you look at all the blue dots represent sites that had severe infections. So it seems like areas that had more severe infections underwent quite a bit of hyposaline stress. Look at the hypersalinity here as a function of time. Now, Labyrinthula does not do well under hyposaline conditions. But again, if you think about the history of the plant, right? If you've got this plant that is lasting and succeeding through a number of years, it's being exposed to the hyposaline stress, and maybe after time, it starts to take the toll on the plant. Zooming in on a narrower time point, when Paige collected, this is going back in 5/30/15, let's look at the salinity, in the month leading up to that. That site Joe, the one that we saw the most severe cases of infection, took a little dip here in salinity. So once again, maybe hyposaline conditions triggered something. And so it seems that, at the very least, salinity certainly has something to do with this. And where we're going with this is it seems that smaller plants are in the east. They've got stronger immune responses, so they have more of a resistance. If we jump down here, they're capable of mounting a response that actively limits the pathogen advancement. Whereas the plants in the central and more so western part of Florida Bay and those basins, they're bigger, they're badder, they're tougher, they're more tolerant. They don't have a strong immune response, but it's like a little gnat landing on a big bodybuilder. It doesn't matter, they can deal with it, So intolerance, they minimize the impact of infection on fitness without specifically reducing pathogen load in the host tissue. And so there's trade-offs between these two disease strategies. You don't have to be one or the other. They're not mutually exclusive. But this is the first time anyone's kind of looked into tolerance versus resistance in seagrasses. And I think we're going to follow up on this in an ecological perspective and kind of dig into this a little bit deeper. Okay, last thing I wanted to tell you, if you're still there. Now that we have this molecular toolkit, we wanted to revisit the Florida Keys and bring that back. And so, with additional FIO cruises, we went back in 2016 and 2018. I'm not going to jump into all the data, but what I can tell you is, things move around and they're very dynamic. If you look at certain sites, like in the Marquesas, 2016, everybody was infected. You come back 2 years later, only 16% prevalence. what happened? And then of course COVID happened, and so that was that. But we want to go back to these sites and start to take a deeper look at what's going on in the Florida Keys and the dry tortugas and the Marquesas. Now, the dry tortugas, beautiful place, and it certainly makes a good excuse to go there to study seagrass disease. Something fishy was going on the last time we were there. A number of the seagrasses, when you looked at them, you saw these lesions. And the lesions are a little bit different than what you see with typical wasting disease. First of all, you only find these lesions on the very youngest leaf, which is not what you find with elsewhere. And when you look at it, you kind of find this horizontal banding. Maybe you can see that on your computers as well, which is kind of weird. And then Labyrinthula, sure enough, was there. Under a compound microscope, it looks like Labyrinthula, but macroscopically, under a dissecting scope, this is Labyrinthula, but they form these really weird esoteric clumps. They undergo this odd, this aggregation pattern that we've never seen before. And if you look down below, this is what typical wasting disease looks like, where you see these brown, black necrotic lesions. This is what we're calling early rank disease, these young blades. Look how it's kind of like a smoother brown kind of blotch. And also we did some experiments on the ship where we took these infected individuals, and on the ship, we took healthy individuals from the same site. We infected them, and 100% transmission. It was quite infectious, whatever this is. Going back to the lab at UNF, we can find all these holes within the cells of the seagrass, the Thalassia. Using SEM, you see a lot of hyphae, this fungal hyphae, which may or may not have something to do with this disease. Zooming in a bit further, there's hyphae, there's co-occurring bacteria. Again, may or may not have something to do with it. Viruses could be present. That's why we want to talk to Maya some more. What are the role of viruses? It's really weird. It looks like also you find these fungal-like conidia, little spores, life history components of fungi. Going back out to fluorescence, here's Labyrinthula. We can see fluorescence, but again, all kinds of fungal-like characters. So the question is, what's going on here? Is it more than just Labyrinthula? Do you have a consortium of microbes contributing to this disease? So we want to certainly look into that a bit more as well. And really, that's probably a good stopping point for me. I mean, that's currently where we are, and we're just kind of waiting for the world to heal a little bit so we can get back out there and resume. But with that, I certainly would like to acknowledge my collaborators, Dan Martin, who's, like I said, the Labyrinthula expert, Brad Furman, who's out there, very excited to work with you. Katrina Lohan is our collaborator at the Smithsonian, and a number of other people as well. And in yellow, these are all the students that just kind of, did all the work here for the most part. Funding agencies, a lot of this was funded internally. Ship time, couldn't have done it without the ship time. And then additional funding came from the Coastal Conservation Association of Florida. So with that, if anyone's still awake, I'm sorry, it was just nonstop. I thank you for your attention, and I'll be happy to entertain any questions you may have.
Thanks, Cliff. That was fantastic. And I promise you we're all awake. That was really exciting stuff. So we typically start with questions from our students. So I see Jennifer Freeland has her hand up, so I'm going to call on her, but other students, feel free to put your hands up. Jen.
Hi.
Hi.
Awesome talk. Really interesting. I was wondering, with the work going down and happening in Florida Bay, has there been any thought or correlation perhaps to storm events, storm surges, when it comes to hyposaline conditions? Just a thought.
Right, exactly. I don't know. It's a great question. We actually, I'm losing my mind. What was the hurricane that passed through there about 3 years ago? Was that Irma?
I think it was Irma.
Yeah, we put it in an SF, kind of those, those fast action responses to address exactly what you're commenting on. It wasn't funded, but no, that's a big question. Exactly. How do storm events relate to this in terms of introducing freshwater pulses? Also, storms are disturbance. So in disease ecology, that's really interesting. What happens when you tear up the entire environment and you start from kind of ground zero? We can look at the transmission and kind of go from there. But to answer your question, we really don't know much about storm events and how that relates to Labyrinthula disease. Big unknown that can join the list of unknowns.
It's really fascinating. Thank you so much.
Thanks.
Thanks. B?
Hello. We met in the student meeting. Thanks again for that.
Yeah.
I was just wondering about, again, back to Florida Bay. Your talk was really interesting, and I'm wondering, what do the patches of seagrass look like in the areas that you found that are more affected? Are they not your typical dense habitat, or are they pretty patchy? Are the edges more variable? What does that look like?
Sure. And to be honest with you, I was not there when samples were collected, and I'm trying to do my best not to kind of bounce anything back to Brad, who has like strong expertise in Florida Bay. Brad, if you want to jump in, if you're still here, you can certainly comment on that. But I don't personally know what those specific environments look like, the little components within the bay, the subbasins, how their density plays out, or what's going on.
Okay, cool. Yeah, it's interesting to see the hyposaline connection. Brad is actually a collaborator on my grant, so hi, Brad. Thanks for your help.
Brad is an encyclopedia.
Absolutely. Okay, we'll open up questions to anybody who's got them. Chris.
Hi, Cliff. Great talk. Really enjoyed it. I'm wondering, and this question is sort of related to what B just asked, and so not to push you back on the spot again, but what do you know about the vectors of transmission? Is it and this goes into the density of seagrass. So do you see it transmitted between blades of seagrass that are near each other? And I think about seagrass environments as being, you can have these large stretches of continuous seagrass, and then you can have very, patchy environments. And so I'm just wondering how those dynamics kind of play out.
Sure. So yeah, definitely blade to blade contact is definitely a major source of transmission. And that's what we've mostly studied. Are there other vectors? Yes, we've been able to isolate Labyrinthula from sargassum, from just detritus. And that's this is going back some years ago, but we've never followed up. We wanted to, but that it's still out there as well. What are the transmission modes? We just don't know.
Very cool. It's wide open then, isn't it?
It's wide open, exactly. And I would assume, right, more dense environments are likely more prone to a die-off. Exactly.
Yeah. Thank you.
All right, I'm gonna jump in and ask one. I actually have many, but in terms of these, I think your infection experiments where you did it, with past stress exposure versus current stress exposure are fascinating. So if you assume that the Labby and the Thalassia were coexisting and they got hit with the same stress and then it got removed, who would recover faster?
The Labyrinthula, probably. It's a fast-growing, eukaryotic microbe. So that's the interesting component. So I bet, the Labyrinthula can bounce back faster and take advantage of that situation.
Fascinating. Great. Okay. Well, I don't see any more hands, so I will ask you in 1 second to unmute, and thank Cliff with all of our claps. But first, I know Amelia wants to talk to you guys just briefly and make an announcement for next week's panel. I'm afraid if we clap, people will start leaving. So take it away.
So thank you, Cliff. As usual, I actually understood something from your talk, so that was really cool. You and Laura do a great job at that. So very cool. I don't have any questions, but what I do want to do is announce to everybody who's still here, and thank you for being here, that we are going to have a special event for next week's seminar, and it's going to involve you guys doing a little bit of work over the next week. What we're going to do is we're going to host a viewing of Picture a Scientist, which is a movie about women in science. And then we are going to host a panel during seminar next week to discuss some of the issues in the movie. The movie is going to be available probably from Monday to next Saturday. And you will be sent a link out. We were hoping to get it today, but the filmmaker seems to be a little bit delayed. So We will get you a link with the promo material for the film. You'll be able to watch it at your leisure before next Friday's panel. So I hope to see you guys all there for something different and really, really important. So thank you for letting me do the announcement. Great job, great talk, Cliff.
Absolutely. So thanks so much, Cliff, and everybody feel free to unmute yourself and give a round of applause.
Great, thank you so much.
Okay, thank you. I enjoyed it. Thank you very much for listening.
Thank you.
Thanks, Cliff. Have a good weekend.
Okay, you too. Have a good weekend, everyone.